Hello, everyone. Welcome to our week 13 lecture of our MCAT question-based review course. This lecture will be on the musculoskeletal system, the integumentary system, and action potentials. So I will be going over 8 questions, all based on MCAT content, and then I will talk about the concepts that underlie those questions. Here I have our GroupMe linked, like always. This is our Instagram handle as well. This is the link to our YouTube channel where you can find all of the videos in the question-based review course and the other videos of content that we do have. And we'll have 2 lectures a week. We have 2 more weeks of the program. We'll have a Sunday recorded release at 8:30 and Monday recorded release at 8:30 as well. So we will begin with our first question here on the muscular system. So when I go to the next slide, you can pause the video, go over it yourself, get the answer, and then unpause the video when you are ready to hear the explanation and the answer. All right, so question 1 here. This states, which of the following describes differences between smooth muscle and skeletal muscle? So because this is a Roman numeral question, I will go over I will review the answer choice that comes up exactly twice. So in this case, I can review Roman numeral 2 or Roman numeral 3. I'll choose to do Roman numeral 2 first. So Roman numeral 2 states smooth muscles do not have actin and myosin, while skeletal muscles do. So we know that's wrong, as both smooth muscle and skeletal muscles have actin and myosin, and those slide over each other like we will talk about shortly. So we can cross that off, as we know that is not an actual difference between them. So I can cross off B, and I can also cross off D. So now I can evaluate Roman numeral 1, or I can evaluate Roman numeral 3. I'll just go ahead and choose to do 1. So smooth muscles do not have striations, while skeletal muscles do. We know this is correct, so that is a difference. And now let's evaluate 3. So 3 states smooth muscles are controlled by the autonomic nervous system, while skeletal muscles are controlled by the somatic nervous system. That is also right. So our answer here is C. So here we have our types of muscles. So first we'll go over skeletal muscle. So skeletal muscles are responsible for voluntary movements. They appear striated under a microscope, and they also are multinucleated. We have slow twitch fibers for skeletal muscles. You can call them also type 1 or red oxidative fibers. We also have fast twitch fibers, type 2. You can also call them white fibers. Those are broken down further into type 2A and type 2B. So when referring back to slow twitch fibers, they had a lot of myoglobin. So that's why they're called, they can also be called red oxidative fibers. Myoglobin is red, and that's when you see that. They also have a lot of mitochondria, so they can generate a lot of energy for a while. And they're less likely, and they have a lot of blood perfusion around them. This makes them less likely to fatigue, and they can sustain contractions for a longer period of time. These muscles are responsible for physical activity over a long period of time. So fast twitch fibers, on the other hand, they have less mitochondria and less myoglobin. So they appear white under a microscope, so we call them white fibers. So they're used for a less period of time, but what they do is that they contract faster. So now referring to the different types, type 2A fibers cannot maintain activity for as long as slow twitch fibers, but they far exceed that type of the fibers of type 2A fibers. So type 2A fibers generate a lot of force, but for a short period of time. When we're looking at this graph that I have, or this chart, sorry, that I have here, you can see the differences between them. this is a good graph to refer back to. So now moving on to smooth muscles, these are responsible for involuntary movements. They exist internally, such as in the respiratory system, digestive tract, blood vessels, and other internal organ systems. Now, moving on to cardiac muscles, these are responsible for involuntary contractions of the heart. They're innervated by the autonomic nervous system, as smooth muscles also are, but they have myogenic activity, which means that they can contract without the input of the nervous system. And then in terms of the heart contracting, that starts at the SA node, and that is what is that has myogenic activity. It doesn't need to be controlled by the autonomic nervous system. And so starting at the SA node, the myocytes are depolarized. These are the myocytes are also muscle cells. This spreads to the AV node, and then the bundle of His, and then the Purkinje finers ultimately. Each cardiac muscle cell is connected by intercalated discs, and depolarization happens quickly due to gap junctions. These allow the electrical current to flow directly into adjacent myocytes. So now our second question here, this is going to be specifically on skeletal muscle. All right. So the question states, during muscle contraction, what causes the muscle to relax? So I will think about what that is before evaluating each individual answer choice on the MCAT. So going over each one now, ATP detaching from the myosin head, or B, ATP attaching to the myosin head. So the answer is actually B when just evaluating those. ATP hydrolyzation also happens, and ADP and inorganic phosphate also attached to the myosin head. Those do happen, but that is not the reason for the muscle to relax. So the answer here is B. If you didn't get this question right, that's okay. We will go over the sliding filament theory, and that's just the theory of how muscle contracts. All right. So looking at skeletal muscle specifically, we have here the epimysium. This is the sheath that surrounds the entire muscle. Going a little bit deeper, each muscle is composed of multiple fascicles. So that's this here. And each fascicle is surrounded by a perimysium. Inside of each fascicle are multiple muscle fibers, which are the skeletal muscle cells themselves. This is surrounded by an endomysium. So that is, you can see here, so each one here is surrounded by the endomysium. Going a little bit deeper, the cell membrane of the muscle cell is called the sarcolemma. So that's this here. And then inside of each muscle cell, we have a myofibril, and these are composed of sarcomeres, which is what you see here. So sarcomeres are what the basic contractile units of muscle cells are. They're composed of thick filaments, which is the myosin here. They're also composed of thin filaments, which is actin. Each sarcomere is connected to each other in a line. So down the myofibril here, they're connected this way at the Z line. Z line to Z line is one sarcomere. The H zone is what you can see here. This is the portion that is only composed of myosin. The I band is the portion that is only composed of actin. And the A band is the entire myosin filament. So in the sliding filament theory during muscle contraction, the A band is not going to change size, while the H zone, the I band will change size. And then the M line here is not shown in this diagram, but this is just the midline of each sarcomere. Okay. So now we'll talk about the impulse transmission at the neuromuscular junction. So what this is specifically, it's the synapse between the axon terminus of a neuron and a muscle cell. So we'll talk about action potentials and synaptic transmission a little later on in this lecture. So if this doesn't completely make sense, I would encourage you to come back to this slide and review all of this. So at the neuromuscular junction, this is where acetylcholine, a neurotransmitter, is released and ends up causing muscle contraction. These here are the steps. So first off, we have the action potential reaching here. And this causes the voltage gated calcium channels to open. So calcium will rush in. The second step is acetylcholine is then released from the axon terminal here into the synaptic gap or the synaptic cleft. And it's going to bind to ligand-gated sodium channels on the postsynaptic membrane here. That causes the membrane to depolarize. So that means there's more of a positive charge inside the cell here. And there's going to be a threshold. So there's a certain value of being positive enough to cause an action potential. If this does occur, then the depolarization will propagate down the T tubules here, which are just invaginations of the membrane. And once that reaches the sarcoplasmic reticulum, that is going to release calcium, and that allows actin to bind to myosin and then cause muscle contraction. And then the action potential will stop once acetylcholine esterase destroys acetylcholine. Now we'll talk about the sliding filament theory specifically, which is now the next step of what happens in the muscle contraction, looking at the sarcomere specifically. All right, so here we have the sliding filament theory. As you can see, so the different steps here, our first step, as you can see here, so calcium, this is what the SR released out into the muscle cell. And so what happens is that calcium binds to troponin. So troponin then is going to change its conformation, causing tropomyosin, which is this filament here, to move out of the way. So it opens up the myosin binding sites to bind. So the myosin binding sites are on the actin filaments. And then the myosin head in step 2, which is bound to ADP and inorganic phosphate, can bind the myosin binding site on actin, and this causes a cross bridge to form just like this. Now, in the next step here, the myosin head moves into a low energy position, in this step here, the myosin head is going to move into a low energy position, and it's going to pull the actin chain towards the center of the sarcomere. And this is called the power stroke, or you can see here also called the working stroke. And at this time, ADP and inorganic phosphate is also released. A new molecule of ATP in the next step here, right here, will bind the myosin head. This allows myosin to be released from actin. This is what happens in rigor mortis when if you're looking at a dead body and the muscles seem contracted, it's because no ATP can come in and bind and cause the muscle to relax. And then our last step here is ATP hydrolysis into ADP and inorganic phosphate. This will cock the myosin head into a high energy position and allow the cycle to begin all over again. All right, so question 3 here. Now we're going to move on to the skeletal system. All right, so first question, what is the function of red bone marrow? Is it A, to drain lymph, B, to form red blood cells, C, to form spicules, or D, to make adipose tissue? So the answer is going to be to form red blood cells or erythrocytes. So that is what the function of red bone marrow is. All right, so now we'll look at the structure and function of the skeletal system. So we have flat bones, which you can think of being the ribs, scapula, other bones like that. Or we have long bones, which are the femur, humerus, also bones of your fingers. Don't seem that long, but also are long bones. Now looking at the structure specifically, we have the diaphysis, which is the long part here. It's the main shaft, also contains the medullary canal, which has bone marrow. There is yellow marrow in this area in long bones, red marrow where blood cells are produced, like we just discussed in the previous question, is present in the spongy bone or the cancellous bone in flat bones. So the metaphysis, this is this part. So this is kind of at the junction between the epiphysis and the diaphysis. It's where the bone begins to flare, as you can see. This is also where ligaments and tendons attach. Then we have the epiphysis. So that is going to be the actual flared part of bone. Here now looking even farther at the microscopic structure of the compact bone, which is this part of the bone here. So we have the periosteum. So this is what surrounds the bone here. As you can see, this is the periosteum. Looking further, this is around here. You have osteons, which are these little units here. These are the basic unit of bones. At the center of osteons, you have the Haversian canals. So that's here. This contains lymph vessels, blood vessels, and nerves as well that run down the bone. And we also have the Volkmann canal. These run perpendicularly to osteons, as you can see here. All right, so now the function of the skeletal system. The main functions of the skeletal system are 1, to support the body, 2, to provide the structure for movement, to protect internal organs, to store calcium, and to synthesize red blood cells, white blood cells, and platelets as well. All right, so question 4 here, also on the skeletal system. All right, so question 4 states, an infant's skull is soft due to areas of connective tissue. However, the connective tissue will transform into bone through the process of A, endochondral ossification, B, periosteal ossification, C, intramembranous ossification, or D, intramembranous ossification. So first, I can cross off C. Sorry, C is supposed to be intermembranous ossification. I'll change that in the slide decks. So if C is intermembranous ossification, then we can cross that off, and we can also cross off B, periosteal ossification, as those both are not forms of ossification. We can also cross off A, endochondral ossification, and so we know our answer is intramembranous ossification. We will talk about what both endochondral ossification and intramembranous ossification is shortly. All right, so other types of tissue of the skeletal system. So we have ligaments. This connects bone to other bone. Tendons connect muscle to bone. Cartilage. This is a strong but flexible extracellular tissue. Cartilage is avascular, so that's why it takes a long time for cartilage to heal. It is also a neural meaning it doesn't have any innervation by any nerves. The types of cartilage we have is hyaline cartilage. This is strong and sort of flexible. Some examples are the lining of trachea and articular cartilage that is at the end of joints. We have elastic cartilage. This is strong and a lot more flexible than hyaline cartilage, like what the epiglottis is made out of. And we also have fibrous cartilage. This is very strong and rigid. This is cartilage that doesn't really move too much, like the cartilage at the pubic symphysis. So now here's where we'll talk about bone growth and remodeling, specifically endochondral ossification, intramembranous ossification. So most bone growth actually does occur by endochondral ossification. So this happens when hyaline cartilage is transformed into bone during fetal development. And intramembranous ossification is when undifferentiated tissue, which is called mesenchyme, is then transformed into bone. So that was in that previous question. That's why the answer was intramembranous ossification, because that mesenchyme will be transformed into bone in an infant's skull shortly after that they are born. So specifically growth of long bones, this happens between the epiphysis and the diaphysis. This occurs at the epiphyseal plate. Here is where chondrocytes divide, forcing the diaphysis and the epiphysis apart. Chondrocytes form cartilage, and then cartilage then turns into bone. Once chondrocytes form cartilage and bone growth occurs, that happens when the rate of ossification of cartilage is faster than chondrocytes dividing. And at the end of growth, the diaphysis and the epiphysis fuse, and this forms an epiphyseal line. So there's an actual line that you can see when you are done growing. That is why when you look at x-rays for individuals that are done growing, that's how you can tell that they're done growing, because you can see this line. So talking specifically about remodeling of bone, this occurs by osteoblasts when they lay down collagen and hydroxyapatite, and osteoclasts break down bone. This constant formation and destruction of bone happens a lot in cancellous bone due to pressure when we walk, for example. And then hormones that we have involved, we have parathyroid hormone. This stimulates osteoclast activity. Calcitriol also does this, while calcitonin inhibits osteoclast activity. All right, so now we will move on here to the integumentary system. All right, so the question states, a laceration that causes bleeding has definitively cut through which layers of the skin? So here I will evaluate, I have 2 or 3, Roman numeral 2 or Roman numeral 3 that show up twice. I can evaluate either, but I will choose to evaluate 2. So if I have a cut and I notice blood, it has to have hit the dermis, because this is where blood vessels exist. So I know that 2 is right. So I can cross out A and I can cross out C. I also know that 1 has to be right, as the epidermis is the top layer. And so if it hit the dermis, then it must have hit the epidermis. But it hit the hypodermis. This we cannot be definitively sure of, because we know blood vessels are in the dermis, but we don't have enough information to say if it's also if the laceration went through and hit the hypodermis. So the answer here is B. So 1 and 2, Roman numerals 1 and 2 are correct. So now I'll talk about structure of the skin. The function of the skin is to protect pathogens, as it is a large part of the non-specific immune system. It also regulates body temperature and prevents excessive water evaporation. So the top layer, the epidermis, is composed of a few layers. So we have the stratum corneum is the top part, the stratum lucidium. This is the next layer, which is actually only present in thick skin, like the palms of the hand or the soles of the feet. You have then the stratum granulosum, and then the stratum spinosum, and then lastly the stratum basale. A good way to remember these layers is the mnemonic "come, let's get sunburn." So "come" is for C for corneum, L for lucidium, G for granulosum, S for spinosum, and B for basale. So the dermis is the middle layer. So as we said, it contains blood vessels, also contains sensory receptors, hair follicles, sebaceous glands, sudoriferous glands. And it's also made up of the papillary layer and the reticular layer. The papillary layer is more of loose connective tissue, while the reticular layer is denser connective tissue. We have the hypodermis. This is loose connective tissue that connects the skin to the body. Also contains fat and fibrous tissue. Also going back to the epidermis, so keratin, I wanted to mention what the function of these are. So keratin is what helps make the skin waterproof. And melanin prevents against UV damage to underlying tissues. Langerhans cells, they're present in the stratum spinosum actually, and they're part of the non-specific immune system. All right. So question six, also on the integumentary system. All right. So first question, question number 6 states, which of the following is not a means that the human body uses to expel heat in warm weather? So we have A, sweat, B, vasodilation, C, panting, or D, active sweat glands. So we know that C is wrong, panting, because that is not a means that humans use to control their temperature in hot situations. This is what animals like dogs do. Humans, however, don't do this. Dogs do that because they don't have as many sweat glands as humans do, so they have to pant to expel heat. We can sweat. Our sweat glands become activated in warm weather, and then our blood vessels also dilate. All right, so we'll talk a little bit more about thermoregulation in the human body. So ways that the skin retains heat. So the skin insulates. The fat that's in the hypodermis that helps with this insulation. We have white and we have brown fat. So brown fat actually releases a lot of heat because the electron transport chain in brown fat is not very efficient. We also have piloerection. So the muscle contracts here, and that causes hairs to stand up when we get cold. And then also the constriction of blood vessels retains heat, so because it prevents losing heat from conduction. So when the body wants to dissipate heat, on the other hand, sweat glands become active. Sweat can then evaporate from the skin, and then also blood vessels dilate in the dermis due to, or in order to stimulate heat loss. All right, so now we will move on to action potentials. All right, so question 7. What is responsible for restoring and maintaining the resting potential of neurons' membranes? So we have A, the sodium-potassium ATPase, B, voltage-gated potassium channels, C, voltage-gated sodium channels, or D, ligand-gated calcium channels. So the answer here is important to know for the MCAT is A, the sodium-potassium ATPase. So this is what maintains and restores the resting membrane potential in neurons. We will talk about this a little bit further in our discussion now. So first we'll go on and talk about the structure of neurons. So we have the cell body here, or the soma. This contains the nucleus. Dendrites are projections from the soma here, and this is where we receive information in the form of chemical signal. It's then converted into an electrical signal that travels from the cell body down the axon into the axon terminals. Different neurons can have multiple dendrites, cell bodies in different positions, but we always have one axon. The myelin sheaths here, these speed up they speed up conduction of the action potentials. In the CNS, they're produced by oligodendrocytes, and in the PNS, they're produced by Schwann cells. However, we'll talk about a little bit more now about how the electrical signal actually is transferred down from the cell body, from the dendrites to the cell body through the axon. So action potentials, what they are is that they're areas of depolarization on the membrane that carry this information through a neuron. So as you can see here on step 5, right here, the resting membrane potential on for a neuron is about -70 millivolts. If we have a stimulus that causes the membrane potential to reach the threshold, that occurs at -50 millivolts, and this is when an action potential will occur. If it doesn't reach -50 millivolts, then the action potential will not happen. There's nothing in between. It either happens or it doesn't. This is called the all or nothing principle. So let's say that we do reach this -50 millivolts of the plasma membrane voltage, then depolarization will occur. So when that happens, sodium channels are open. Sodium flows down its concentration gradient into the cell. This occurs until the membrane potential is about +35 millivolts. And then as you can see here, the way that a action potential is propagated is because some of the ions flow down the interior of the axon, causing the membrane potential to increase. So it starts to go this way, and then it propagates that way as well. However, going back to this part, at the +35 millivolts, what happens is that the sodium channels, they are inactive, and then the potassium channels then open. So potassium ions then flow down its concentration gradient out of the neuron, and potassium channels are also slow to close. So that's why you see this hyperpolarization here. So the potential actually gets all the way down to about -90 millivolts. The point of this hyperpolarization and the potassium ions to be slow to close is that it creates a refractory period where the region of the membrane cannot fire an action potential anymore. This forces the only the area only to the right side on the axon, in order to actually have an action potential. So that means it will only propagate that way. Potassium leach channels, on the other hand, and the sodium potassium ATPase allow the membrane potential to return to normal, as we talked about in the previous question, and the sodium ions become active again, so another potential can then later occur. So also about the sodium potassium ATPase, important thing to know is that it uses ATP to pump 3 sodium against its concentration gradient, and also uses 2 potassium, or it spits 2 potassiums against its concentration gradient, which is into the cell. So that is important to know about the sodium potassium ATPase. All right, so I will then move on here to the last question, question 8. All right, so question 8 states, if acetylcholine is released from the presynaptic neuron, but remains in the synaptic cleft due to a loss of function mutation in the gene that codes for acetylcholine esterase, then what is the postsynaptic membrane? Then the postsynaptic membrane will blank. So will it A, spontaneously depolarize, B, spontaneously hyperpolarize, C, be depolarized longer with each action potential, or be depolarized by action potentials more frequently? So what happens is that if we don't have acetylcholine esterase, then the ion channels in the postsynaptic membrane will be open for longer, because acetylcholine will be in the postsynaptic cleft for longer. That will make depolarization last longer. A and B, about it spontaneously being depolarized or hyperpolarized, doesn't make sense, so we can cross those off. And for C and D, it is going to be C. It's just depolarized for longer. It won't happen more frequently. Just you have acetylcholine there binding to receptors, so we know that it's going to last longer. All right, so we'll talk about now synaptic transmission. So once the electrical signal then reaches the synaptic knob, this is what occurs, and now we go from an electrical signal now to a chemical signal, and to talk to either the organ system that it is going to that the organ system that it touches, or the other neuron. So talking about each step. So first, what happens is that we're going to have voltage gated calcium channels that open. So this occurs once the nerve impulse here reaches the synaptic knob. Calcium is then going to influx into the cell. And then what occurs, we have these vesicles that contain neurotransmitters. Then it's going to form here with the membrane and exocytosize the neurotransmitters that are inside. They will be then released into the synaptic cleft here. Neurotransmitters will then bind to the ligand-gated ion channels on the postsynaptic membrane. The ions, as you can see here, will enter the cell, and polarization of the membrane either increases or decreases. This last point here is important because not all neurotransmitters have an excitatory effect. They also can have an inhibitory effect. So if it has an inhibitory effect, that is going to be called an inhibitory neurotransmitter. While if it causes an excitatory effect, it's going to be a excitatory neurotransmitter. But an important point also is that the receptor on the postsynaptic cell is what causes the effect on the cell, not actually the neurotransmitter itself. I hope this discussion here clears up a little bit about the neuromuscular junction as well. So I would encourage you to go back to that slide about the neuromuscular at the neuromuscular junction if you were a little confused about that. But that is it for tonight. Good luck studying. And again, feel free to reach out to me by email or potentially through GroupMe as well. All right. Well, have a good night. We'll see you next week.